Imagine you're the Secretary of Health to the US government and you're extremely worried about the fact that in the past year, over 600,000 people have died of cancer in the US. But now you're presented with this encouraging data from the CDC that if diagnosed in the early stages, the 5-year survival rate for most types of cancer is over 90%. But now you get tense again because in the early stages, most cancers don't have any obvious symptoms. And to screen the entire population using existing invasive techniques like biopsy is practically impossible. So you ask yourself, could it be possible to diagnose cancer from saliva just like we've done for COVID? Herein comes my PhD, where I utilize the power of material science and machine learning to tackle this problem. But first we ask, what should we look for in saliva? The answer is exosomes. Exosomes are particles released by all the cells in our body, including disease cells. What's fascinating about exosomes is that they act as a screenshot of the cells that produce them. So an exosome from a cancer cell contains the markers of the disease itself. What's more, exosomes are extremely stable in our blood supply, and some of them do end up in our saliva. Now this sounds perfect, so why haven't we diagnosed cancer from saliva already? That's because exosomes are extremely tiny. They are over 10,000 times smaller than the thickness of a strand of hair. So to search for them among the billions of other particles in a saliva sample requires an extremely sensitive technique, which is a key technological challenge. To this end, in my PhD, I work on a technique called surface enhanced Raman spectroscopy, or SERS. Now SERS utilizes the unique interaction of lasers with metal nanostructures to help detect tiny particles. The lasers interact with the chemical bonds in these particles to give very specific signals called Raman signals. But these signals happen to be weak, so we make metal nanostructures to help enhance these signals by over 1,000,000 times. This helps detect the tiniest of particles in saliva, including exosomes. Now in my work, I collect a data set of healthy and unhealthy samples, which I use to train machine learning models for the purpose of diagnosis. In fact, our group has used this for gastric cancer diagnosis, and we already have an accuracy of over 70%. My PhD thesis focuses on addressing the challenges like higher accuracy, collection time, to help bring this closer to clinical trials. And my dream is to see this technology enable early, non-invasive, and more accessible diagnosis of cancer to save lives. Thank you.